Morning, good afternoon, good evening, depending from your regions. I'm happy to welcome you to this webinar. My name is Casimiro Vizzini, and I'm speaking from UNESCO, where I serve as coordinator of the partnership with the Human Cell Atlas. It is a pleasure to welcome you to the first session of this series towards a human exposome atlas. Before we begin, a few logistical notes so that the session runs smoothly. The webinar is being recorded. You're invited to submit questions at any time through the Zoom Q&A window. We will collect them throughout the session and bring them into the moderated discussion later. The chat will remain reserved for announcements from the organizers and including links to documents and follow-up materials. We kindly ask all the speakers to keep an eye on the time and remain within the allocations indicated in the agenda. This will allow us to preserve space for the collective exchange at the end. Thank you again for joining us. And now, Bridget, if you want to give some more information.
Hi, yeah. Please, if you have any questions throughout, please populate those in the Q&A, and we can hand that off to Fenna now.
Thank you, Bridget. I think all the other speakers, we remain with our camera off and our mic off during the others' intervention, so we can then open. So let me start.
Yes. Okay, sorry.
Yeah, let me start saying that I would like to begin recalling why this conversation matters today. Across the world, we are confronted with growing evidence that environmental exposures influence health outcomes from early life to aging. At the same time, single-cell science has given us the ability to understand how these exposures leave signatures in tissues and organs. When we bring these two perspectives together, we move closer to a knowledge base that can support earlier detection, better prevention, and more equitable policies for population health. This webinar series is therefore more than a scientific exchange. It reflects a broader movement toward the integrated approaches for understanding the environmental determinants of health. It also builds on emerging recognition within the scientific community and with governments that exposure science and single-cell mapping are complementary. Together, they can produce insights that were simply not accessible a few years ago. The challenge now is to connect communities, harmonize standards, and create shared frameworks that enable data to be used responsibly for the benefit of all. UNESCO's role in this space is anchored in two pillars. The first is our mandate to promote open science as foundation for inclusive and transparent knowledge production. The UNESCO recommendation on open science emphasizes the need for accessible data, interoperable infrastructure, and collaborative research ecosystem, while at the same time protecting personal data, privacy and security, and recognizes indigenous knowledge. This balance is essential for fields that involve sensitive health and genomic information. The recommendations provide guidance on how to keep science as open as possible and as protected as necessary, ensuring that innovation does not come at the expense of ethical or legal responsibilities. The Human Cell Atlas has already demonstrated how global participation and data sharing can be built on robust governance and high standards integrity. The experience will be valuable as the Exposome community expands its activities and strengthens its international collaboration. The second pillar is science diplomacy. The recent UNESCO Global Ministerial Dialogue on Science Diplomacy brought ministers and high-level representatives together to reflect on how international cooperation can remain effective in a rapidly changing world. It was held here in UNESCO earlier this year in March. The dialogue underscored that geopolitical tensions, technological disparities, and unequal science capacities are fragmenting collaboration at a time when global challenges require coordinated action. It also emphasized that science diplomacy is not only about negotiation. It's about creating the conditions for trust, for shared standards, and for informed decision-making based on evidence. Several ministers highlighted the need to protect large-scale research collaborations, to support equitable access to data, and to reinforce scientific infrastructures as global commons. These messages strongly resonate with the work being presented today. In this context, the connection between the human exposome agenda and the Human Cell Atlas, let's say the Global Exposome Forum and the Human Cell Atlas, becomes a concrete example of how science diplomacy, open science, and scientific excellence can reinforce each other. Exposure science requires multi-sectoral data, cross-regional cooperation, and long-term monitoring. Single-cell mapping requires open methods, shared protocols, and inclusive participation to reflect human diversity. When these approaches converge, they offer a pathway toward a human exposome atlas that is scientifically sound, globally representative, and aligned with principles of equity and solidarity. This vision also speaks to the International Decade of Science for Sustainable Development. You can find the link in the chat. The Human Cell Atlas is endorsed by the Decade of Science for Sustainable Development. This decade calls for transformative scientific cooperation guided by societal needs. Environmental exposures are not distributed evenly. Unrepresented communities and low resource settings are often the most affected and the least studied. Building an exposome atlas with global relevance therefore requires capacity building, shared infrastructure, and fair participation from all regions. UNESCO stands ready to support this effort through its programs on open science, ethics, capacity development, and science diplomacy. I would like to welcome all the speakers and participants who are contributing to this session. Your reflection will help us to identify the opportunities for collaboration and the steps needed to build a coordinated approach. I'm confident that the dialogue beginning today will contribute to a common vision for advancing exposome research in a way that is open, ethical, global, and inclusive. Thank you for joining us, and I look forward for the discussion ahead. Fenna, I think the floor is yours.
Yes. Thank you, everybody, for joining us today. And I'm representing the Global Exposome Forum today, and will briefly explain what the Global Exposome Forum is about and how we will join with UNESCO and the Human Cell Atlas towards a Human Exposome Cell Atlas. So 20 years ago, Christopher Wilde coined the concept exposome as a complement to the genome, which means that this is anything but the genome, but it's working together with the genome. So it's the air you breathe, the water you drink, the stress you're perceiving. The Banbury meeting in 2023 was a follow-up meeting that redefined more practically what the exposome is. Briefly, the exposome is posed as an integrated compilation of all the physical, chemical, biological, and psychosocial influences that impact biology. The field of exposomics thus examines these comprehensive and cumulative effects of these factors by integrating the data from interdisciplinary methodologies and data streams to drive discovery. As a complement to the human genome, the human genome was mapped by a massive undertaking called the Human Genome Project. We had several people in the community had a call out for a complementary project, the Human Exposome Project. Last May, we had a summit, a forum, where we brought different stakeholders together to meet in Washington, D.C. to discuss how we would go towards a Human Exposome Project, a complement to the Human Genome Project. This Exposome Moonshot Forum brought together stakeholders from academia, government, patient representatives, policymakers, industry, end users, people that had never really come together to discuss how we practically can translate human exposome towards a human exposome project. The outcome of this Exposome Moonshot Forum is where we had discussed in breakout sessions and panel discussions about the enabling technologies, the big data, psychosocial and ethical aspects, global coordination, and how infrastructure would be led, as well as training the next generation, how to engage and reach out to the community, translate to practice and at the individual and the population level, and how to keep things open, science, harmonized, and fair. The participants of the meeting all signed a declaration, and I have links in the chat soon where you can still sign this declaration towards a global consortium for the human exposome. After the Human Exposome Forum, the Global Exposome Forum was established, which gathers all the global chapters that are working towards this end goal of a Human Exposome project. And this may mean that these are parallel regional human exposome projects, but under the number of a Human Exposome project at large. We're working on a roadmap, which we're presenting at the next conference and in Barcelona, Sitges, and I have a slide on that as well, where we will map out what the steps are needed to reach a human exposome project so that we can complement and interact the data with the Human Genome Project so that we can understand all the social determinants of health, but also the chemical and the exposome, and that contributes to the global health burden. The Global Exposome Forum is established, will be established as follows with a Global Steering Council. We already have an interim executive committee who will oversee the daily logistics, etc., and the governance of this Exposome Forum. There will be working groups, and we have a call out for interest so that you can join one of these working groups to help map out these specific areas, to refine them so that we can work to this Human Exposome Forum, Human Exposome Project, sorry. There are task forces which are tasked with fair data standardization and the economics and the stability of and sustainability of this project, and we'll have self-organized regional chapters to support these efforts globally. And because ethics is very important, we'll have an independent oversight in ethics boards as well. The next conference, as I mentioned, is the Global Exposome Summit. This will be held from the 27th to the 29th of April in 2026 in Sitges near Barcelona, and you can sign up for registration soon. So with that, I would like to conclude, and I hope and look forward to how the Global Exposome Forum can join together with UNESCO and the Human Cell Atlas towards a Human Cell Atlas. Thank you. John, the floor is yours.
Yes, thank you very much, Fenna. My name is John Randell. I'm the Executive Director of the Human Cell Atlas, and I'm very pleased to be here. Really looking forward to the talks and the discussions today. I will begin with a really brief overview of HCA and what our progress has been towards the atlases that we're creating, and then I'll talk a little bit about how the exposome fits into our long-term plans. There we go. So the mission of the Human Cell Atlas since our founding in 2016 has been to create comprehensive reference maps of the types and properties of all human cells as a basis for understanding, diagnosing, monitoring, and treating health and disease. And we are doing this through a set of powerful new technologies that have really been invented in the last 10 or 12 years or so, known as single-cell genomics, as well as spatial biology approaches and a variety of different single-cell and spatial omics. And the key for us is that with around 37 trillion cells in the body, this is obviously a really monumental task. It's one that one laboratory or even a group of laboratories really cannot do alone. And so we are really a global community that got its start as a grassroots effort. And we have now grown to be over 4,000 members around the world, based at nearly 2,000 institutes in 104 countries. So really quite a global effort. And you'll see that represented in some of the talks today. Now, this group or this project is led by an organizing committee, what we call the organizing committee, which is a scientific steering group made up of around 30 scientists from around the world, and it is supported by an executive office of professional staff, which I lead. HCA is organized, a lot of our work is organized around what we call our 18 biological networks. And these are research networks that encompass all the tissues in the human body. They comprise anywhere from 100 to over 600 scientists per network. So these are very large research networks. And they're working collaboratively to develop single-cell atlases covering each of the tissues in the body. So in the first phase of HCA, which is just drawing to a conclusion next year, we are asking each of those research networks to create one or more single cell atlases for their particular tissue, organ, or system of interest. To date, we've approved, or at least we've had at least one atlas approved for 11 of those 18 biological networks, and the remainder will be completed next year. And actually, there are 11 on the left and 8 on the right. So if you add that up, that's 19. And that's because the lung network, which really it really got its start actually at the very beginning of the COVID pandemic, is working on its V2 atlas. And so that just goes to show that these atlases will be iterated over time to add more data and more data modalities and expand diversity. And I'll talk about that in a second. So this really is, I want to emphasize, this is just the first stage of HCA. It's these individual tissue atlases. In the long term, we need to stitch these together to make a whole body atlas and one that really encompasses the entire breadth of human diversity. So our strategic goals for the next few years for what we are calling HCA 2.0 includes many different overlapping realms. One at the bottom there, as I just mentioned, is to do the cross-tissue integration and annotation to create a single whole body reference atlas. In the upper left, we are just starting to layer on spatial information using spatial omics approaches. We are exploring clinical applications in the upper right there. And then sort of at the center of all of it is the development and the training of foundation models to expand our capacity to understand biology. And surrounding all of this is the question of global representation and human diversity. And for us, that really means diversity in all of its forms. Certainly, gender diversity, ancestral diversity are paramount. But going back to our original founding white paper, which was published in 2017, I noticed that from the very beginning, environmental or experiential diversity was also a key component of HCA. So you see this quote here, notably genetic diversity is only one set of factors. We need to consider experiential and environmental diversity to ensure that biological insights will be applicable and the health benefits of this research will be applicable to people everywhere. And so it's really, I really see that this new potential collaboration is a really powerful way to address these new forms of, or new for HCA forms of diversity, and you'll hear some great examples of HCA research in the talk today. To that end, we've created with the Global Exposome Forum a joint registry of researchers who are interested in exposome topics. You can scan the QR code here, and this will be available later. I also want to encourage everybody to to sign up for membership in the Human Cell Atlas. Our membership is open to everybody as long as you support our values and principles. You do not need to be funded by a particular mechanism or even have an active research program in order to be a member. And so I encourage you to sign up, and that way you'll get notices of future events and such as this one. And finally, I want to invite everybody to our next annual meeting. We call it the HCA General Meeting. The next one will be held in Boston next June. It is a coincidence, but I will note that that is also coincides with several World Cup matches, both in North America and in Boston specifically. Also, our HCA Asia Regional Network, which is quite a large meeting with hundreds of scientists attending, will be held in Perth in November of next year. So with that, I will conclude, and I believe I am turning this back. Thank you.
Thanks very much, John, and Fenna, and everyone, Casimiro, for kicking us off. Now we're moving to the science part, so you can all see how this kind of work is going to change the world and what it means and how you can also participate. 4 lightning talks, 2 from Human Cell Atlas, 2 from GEF. 15 minutes each. Encourage the speakers to leave at least a couple of minutes at the end for questions. And everyone, please use the Q&A button in the Zoom to ask your questions. All right, our first speaker for this round is Nicolo Caporle from Human Technopole and University of Milan. Nicolo?
Thanks. Do you see my screen?
Yes.
Great.
Yes.
Thanks a lot for the introduction. It's an honor to be here, and I will show you today the work that we have been doing in the last years to generate and share a molecular cell atlas of endocrine signaling in human neural organoids as a paradigmatic example of a project that show how to integrate global exposome forum and human cell atlas aims. So the overarching aim of our lab is to study the molecular landscape of human neurodevelopment, and in particular how genetic and environmental factors interact in shaping this complex regulation. To do that, we leverage the revolution introduced by induced pluripotent stem cell reprogramming and the differentiation of neural organoids as a way to experimentally model in the lab the early stages of human neurodevelopment and disentangle genetic and environmental factors that can impact on them. In particular, as you can see here, recently the community established frameworks and guidelines on how to properly perform this in vitro disease modeling. Here I would like to mention a very important initiative for the Human Cell Atlas that was released last year and to which we only made a small contribution, where single cell transcriptomes from a lot of different neural organoids protocol were integrated and annotated by mapping them towards human fetal references, and this allowed us to get a better understanding of which cell types are recapitulated in neural organoids and when, also depending on the different protocols that can be used, in particular differentiated guided and unguided protocols. Moreover, we recently spearheaded an innovation for this field by introducing and characterizing different ways to multiplex cortical brain organoids into the dissection of neurodevelopmental trajectories. This is key for us because it allows to differentiate mosaic models, so organoids that that includes multiple individual lines, and this goes towards the communal effort to scale up this system to study neurodevelopmental trajectories for a lot of individuals. Now, going back to the general interest of studying the impact of environmental factors, since 10 years now, we focused our attention on dissecting how endocrine disruptors can impact human neurodevelopment. Endocrine disruptors are chemical compounds that derive from a lot of different sources, and they are widespread in our environment. We are all exposed to them. So our innovation was to tackle this problem by looking at real life. So we leveraged a collaboration with Swedish cohort Selma, that is a longitudinal pregnancy study, where a lot of different endocrine disruptors were measured during pregnancy, and then the children were followed up during their growth. Thanks to biostatistical analysis, we identified a specific mixture of endocrine disruptors, particularly containing bisphenol A, phthalates, and different perfluorinated compounds, as you see, that was synthesized and exposed in the lab. So the key idea was to integrate epidemiological association with experimental dissection to identify real-life relevant mixtures of endocrine disruptors and dissect their impact on neural cells. And thanks to that, we discovered that there is a convergent mechanism between environmental factor and genetic susceptibility in the pathogenesis of neurodevelopmental disorders. So following up on that, thanks to an European project that involved a lot of different partners, in the last 6 years, we established a molecular atlas of hormonal signaling in neurodevelopment through organoid modeling to really map the molecular effect of the perturbation of the different endocrine pathways that we think are relevant for proper neurodevelopment. And in particular, we exposed chronically cortical brain organoids for 50 days from 2 cell lines, one male and one female, derived from healthy donors, to agonist and antagonist of 7 key endocrine pathways, including estrogen, androgen, glucocorticoid, thyroid, retinoic acid, aryl hydrocarbon, and liver X. Following up on this exposure, we performed in parallel bulk and single cell transcriptomic analysis, targeted steroidomics, and immunohistochemistry. So this allowed us to first characterize the transcriptional impact of the perturbation of the different hormonal pathway. And as expected, we observed that all the different perturbations produced an effect. And in particular, across different endocrine axes, the response of the male and female line was similar across the two, or could be different in terms of the response to the hormonal perturbation. So proceeding on that, for each of the pathway, we characterize the specific genes that are significantly altered. This is the example of the effect of retinoic acid agonist, where we saw a strong upregulation of genes related to neural differentiation and a downregulation of genes related to cell divisions. The important message is that for every pathway, we characterize the molecular effect. However, we could leverage this data also to identify novel crosstalks, because we know that endocrine pathways act on a shared landscape. And so by leveraging weighted gene co-expression analysis, we identified in particular these 2 modules of genes that are heavily shared across glucocorticoid, thyroid, and liver X, and are particularly enriched for biological processes that are related to metabolic regulation and protein folding. Thanks to the single cell transcriptomic analysis, we could also identify the different cell types that were present in the neural organoids that we exposed. And after dividing the progenitors and the neurons and as main cell type category of our system, we studied the impact of the hormonal perturbation at this finer level. And in particular, we identified that hormones seem to act at the level of different cell states, such as, for example, the identification of caudalizing progenitors and neurons that are induced by the exposure of retinoic acid, so cells that are more similar to the posterior regions of the brain, or the identification of specific subclass of neurons, such as the one expressing LHX9, which are a developmental state enriched for the organs that that were exposed to aryl hydrocarbon. On top of the molecular characterization, we also performed systematically immunohistochemistry for the organoids. So by identifying and quantifying the number of progenitors and neurons in the different organoids exposed, we could also characterize at the level of proteins and cytoarchitecture the impact of these compounds. Interestingly, since we developed the automatic pipeline to segment the nuclei of different organoids, we now have a lot of data points, millions of nuclei from the organoids that we can analyze in terms of morphometric parameters. And so we also identified that the exposure to hormonal alteration was also impacting on different size-related or shape-related features of the nuclei of the organoids. To conclude, we released this work as a paper on bioRxiv that you can check, where we also defined the hallmarks of the endocrine signaling in neural organoids. And I wanted to highlight that the data sets are published as a collection of the Human Cell Atlas and available for downloading and exploring in the CellXGene portal. And we also released a very detailed GitHub repository where you can find all the data, but also a mining of our results that we did by comparing the molecular and cellular impact we found with the literature knowledge that was already available. As a second part, just a glimpse into what we are doing now, in the sense that in addition to the characterization of the impact of hormonal agonist and antagonist, we also performed a follow-up of our previous studies on endocrine disruptors, this time exposing the organoids to all the single EDCs that are present in the mix that is associated to the clinical outcomes, and also performing a dose response design, which is very important to characterize the impact of these complex chemicals. This is still unpublished, but we identified a strong impact by exposing the organoids to all of these single endocrine disruptors and to the mix. And for every compound, we characterize again the genes that are differentially impacted and their relevance in terms of biological enrichment and disease relevance. Interestingly, since this time we have a dose response design, we are analyzing our data to define dose response patterns in response to the different compounds. And this is a very important approach because it allowed us to identify specific doses, which are defined as point of departure of the response, which are very interesting because you can use this in vitro derived data to reinterrogate epidemiological data to understand the association between specific doses of endocrine disruptors and the clinical impact. To conclude, some insights on what we are doing now, and so our future perspectives is based on this European project that is called REMEND, which aims at building resilience against mental illness, and where we are again collaborating with the epidemiological cohort SELMA and other cohorts to this time leverage the three axes that we are interested in. So stratifying the individuals based on their phenotype, their environmental exposure to EDCs, but also their genetic predispositions. In this way, we could identify subgroups of individuals that are resilient or vulnerable to the exposure to the endocrine disruptors, and we are reprogramming induced pluripotent stem cells and differentiating these organoids from the same individuals. Similar, we are leveraging CRISPR perturbation approaches to disentangle this interesting question of how gene and environment interact for giving the phenotype. And so we are perturbing specific targets with a novel approach that is called CropSec multi. And while we differentiate the neural organoids, we expose the cells with different perturbation to the physiologically relevant endocrine disruptors. And to conclude, we thought to cite this recent review because we try to illustrate how nowadays the single cell level of analysis is the pivotal placeholder for our biological inquiry, because it allow us to understand in parallel the complexity of the brain, but also how different individuals contributing to this diversity. So if we place single cell analysis in the context of this integrative approach from epidemiology to neural organoids modeling, we aim to integrate the global exposome forum with the Human Cell Atlas. And with that, I would like to thank you for your attention and the great colleagues with whom we work every day.
Fantastic. Thanks very much, Nicolo. Maybe time for one lightning answer to one lightning question. How to validate in terms of in vivo brain responses?
How to validate our findings in terms of in vivo evidence?
Yes.
Yeah, so our idea is exactly to follow an iterative approach where we basically start from hypotheses that derive from epidemiological association, and then we go in the lab to test mechanistic insights. Once we identify key genes, thanks to disease modeling in vitro, we can go in different levels to validate them back in the epidemiology. One approach is to check if the doses that are important to elicit the response in vitro are also finding some patterns in other cohorts in vitro from the same, if we have the same chemical compounds that we can use. And the other approach that I think would be very interesting in a setting of global collaborative effort is to start from the gene markers that we can identify by in vitro modeling and start going into the human samples to, for example, look for gene expression biomarkers or DNA methylation biomarkers from human samples, when you have cohorts where you can map both the exposure and the phenotype.
Great preparation for our panel discussion afterwards. I'll hand over to Fenna for our next speaker. Thank you once again, Nicolo.
Thanks a lot.
Yes, thank you. So our next speaker is Dr. Chirag Patel from Harvard Medical School, and his talk will be about interrogating the multi-omic architecture of the exposome and intervention from populations to individuals. Chirag?
Great. Should I go ahead and share?
Yes, please.
Great. So can you all see my screen?
Yes.
Great. Thank you
Yes.
so much, Fenna. When you and the HCA team had and the Exposome Forum had introduced me to this vision, I could not say no to this. This is extremely exciting, and I'm so honored to be a part of at least some of the preliminary planning stages, of which I think is going to be a very important project to help us refine and make more precise some of the estimates we're getting from populations and individual level, blood level data, to eventually get to cells, which I think is a broad and fantastic vision. So as you mentioned earlier today, Fenna, the exposome is this integrated compilation of things that are external and internal to the body that impacts biology outside of genetics. So you mentioned this science paper of which you had participated in to essentially bring together a field of exposomics to discover new factors or mixtures of factors, as the previous speaker, Dr. Nicolo, just mentioned, that impacts disease. This is complementary to our existing ways of studying environment, in that we are now thinking of things across multiple domains of exposures to really get a sense of how all these things combine, iterate, and on top of each other to really impact phenotype. The reason for the season, why we're here, and why I think the HCA is developing the resources. And so our fundamental model, as you know, is that phenotype, including single cell expression, is a function of things that come down from mom and dad, such as our genetic variants, and of course, exposome. But I think that in today's resources, we're really focused on the broader resources that genetics community has brought to bear. So just to give you a sense of this, we have seen multiple impactful papers that have leveraged things such as the HCA to understand the role of individual level variation, genetic variation in expression and biological function. Through these scalable tools such as the genome-wide association studies. So this is just an example of the scale that it has achieved. And we're still trying to achieve that scale on the exposome. And I think that's what the promises of the exposome is, to try to get to multiple factors, essentially beyond lifestyle, beyond endocrine disruptors, to understand really what is the drivers of disease beyond those things that we are candidate factors that we are looking at. So in the genome-wide association world, we figured out what variants and what genes, how to replicate, what are the confounders, and what single-cell experiments would like to bring to bear. And I think what I would hope from our exposome community is we can have a analogous effort to understand what exposures, how to replicate them, what are confounders, and how to bring them into single-cell experimentation. So, not unlike our in the genome world, we're trying to understand different questions in the environmental space with the exposome. So, how much variation do we attribute the exposome to phenotype? This idea of genetics plus gene by environment interactions plus non-shared exposome, which I think single cell atlases will be able to get to, as well as shared exposome. So the idea is larger the proportion, more efficient discovery. And the critical questions we need to ask here are what and how many factors are associated? How do exposures accumulate? Are there multiple factors of small effects, few factors of large effects? And are we thinking about acute or chronic types of phenotypic responses? And this is where I think that the discovery of doing things like an exposure-wide association really come into play. That's what I'd like to illustrate today. So first is when moving from populations down to cells, first of all, I think as also you've mentioned earlier today in the questions, that there's a great deal of heterogeneity when dealing with these different types of scales of data. So here, what we did to try to understand the shared environmental piece is we looked at how individuals share for example, weather, air pollution, and area level demographic information to look at the impact of phenotypic response in the shared environment. So how much variation do we expect the exposome to capture on a population level? So you've probably heard of terms like heritability. This is a similar idea of what we call exposomability, is how much environmental variation left to be explained in phenotype. And to do this, we used a natural experiment of twins in a health claims data set where we had 60,000 twin pairs approximately to look at this, to partition the variation of shared environment genetics on 501 phenotypes. And so what we found was that there's a great heterogeneity of heritability. This is the idea of the total variance explained of G, of total genetics, and about 10% of shared environment. That is to say, if individuals who live in a similar zip code or census tract, they share about 10% of variation on average across these 501 phenotypes. But you see here from the bar charts, which summarize across these different phenotypic groups like metabolic response, endocrine response, that there's a great deal of variation across heritability, and in the green bars or the blue bars, shared environment across these different domains. And so just the gestalt here is that when looking at heritability, shared environment, which is what we call C squared, and total residual variance, which is E squared, that's the red here, there's a lot left to be explained, of which I think the single cell approaches could help us refine that variation. So across 560 phenotypes, we about average 30 to 50% of heritability, 10% on the shared zip code, but a lot left to be explained on an individual level basis, which of course could be random chance, or as single cell experimentalists like to call it, potentially expression due to noise. So what I'd like to illustrate here is that we can look at blood, and the promise of looking is single cells is moving beyond blood. But even with blood, we have a great deal of information that are emerging about phenotypic exposure response. Again, similar to the results that were just presented in the last presentation. So here we took 305 phenotypes such as those that are measuring related to kidney disease, metabolic disease, anthropomorphic measurements, bone-related measurements, blood parameters, and related them all systematically to 619 targeted exposures, things like phthalates, VOCs, hydrocarbons, heavy metals, to run what we called a phenotype by exposure-wide study across these 305 and 619 exposures, which is currently in press. And our hypothesis was that there's a structure behind, there's an architecture behind the exposure and phenotype associations that are diverging from what we find in genetic architecture from genome-wide association studies. And indeed, that's what we found. In fact, if we looked at these 619 exposures and 278 clinical phenotypes, we found a vast array of associations, about 5% at the most stringent bond-forming level across these domains. This is what we call a Manhattan plot for exposome-wide association studies. On the x-axis are the different categories of exposure, and what we're displaying here are the different categories of phenotypes. And you can see here just the vast signals that are emerging across the exposome onto these phenotypes. So the next questions might be is that how do these induce a single cell expression response on the way to inducing clinical variation and change? But I would say that the architecture is very complex, and that's one challenge we'll have to address when going down to the single cell, in that the total variance explained is quite modest. So on the level of the single exposures at a time, we were getting anything from on the median of 0.5% of variance explained all the way up to 5%. So if you're thinking of that red plot, that red figure I showed you earlier, we're just inching towards explaining that vast amount of 60% of variation explained. And it's quite divergent across these different domains of exposures. That's what's being shown on the right-hand side on the average. So things like heavy metals, dietary biomarkers seem to play a larger role, but still a lot more left to be explained in phenotypic variation. So we got a median of about 1% in R squared in the interquartile range, about 0 to 2% of variance explained. And if you compare that to the genetics, it's quite comparable, as a matter of fact. So what we did was here is we looked at the R squareds, or the variance explained for each of these phenotypes, these 200 plus phenotypes that we examined, and we asked how much incremental variance explained, similar to that last plot I showed you, that each of these exposures are adding. And that's what you see on the black points, that lift that you get from a base model that includes age, sex, ethnicity, socioeconomic status. What's in the red here are of interest of like 20 or so exposures for each of the different phenotypes. If you start adding in, you have variance explains that are lifted even a bit further, up to 60%. But most are indeed still modest, on the average of median of 3.5% when adding 20 exposures. So here, there's two things that I'd like to evoke here, is that once we start to go exposome wide, that is using new technologies to measure the exposome, which I feel are super exciting to add to single cell experiments, we'd expect this variation to increase. And what phenotypes could be explained by these exposures are going to be super interesting, especially when we get to the single cell level. So here's the type of structure you might see in these data. This is a heat map of the exposure phenotype relationships. And here, just to appreciate that there's a vast differences between how you measure between a dietary biomarker and an interview, or across the different domains. So you see things like hydrocarbons have a persistent response across multiple domains, some sort of like environmental like pleiotropy, and others remain strong in specific phenotypic domains, such as the VOCs and the blood parameters, or the organochlorines and the metabolic or the anthropometric parameters. So as I was saying earlier, the technologies to measure on the individual level are getting better and better and better of exposures. So and that's what's exciting about this forum here. If the exposome is causal, then it must perturb biological pathways. How and in what tissue and cells beyond the blood? So here's a fantastic perspective from a colleague of mine, Peng Gao, who wrote about scaling up exposure measurements using mass spectrometry on the single cell level, which I think is super exciting and relevant here. these are clinically relevant to go to the organ or single cell level. Here's a case study to show how Parkinsonism was induced by heavy metal exposure, specifically manganese exposure in individuals with who deal with welding. And this is an example that toxicologists would probably know, but I think this is very important to take forward when studying single cells of different organs, is how exposures impact cells tissue-wide to induce their response. We're trying to get to this using things like MRI and biobank scale data. Here we predicted age using MRIs to understand what are the biological structural changes in these different organs and then relate that to exposures. That was a paper a few years ago. So we did an XWAS on that biological age predictor, and we found things like alcohol, sleep, correlated with an abdominal age as structural age as reflected by the MRIs. But what I'd like to leave you with is this super exciting project we've been also leading with these biobank samples, is looking at the omics as a mediator. So you could think of single cell expression as a mediator, as an integrator of both genetic and exposure signals beyond potentially random bursts. So here what we did, Shaxon Isaac and my team integrated UK Biobank exposures and genetics and looked at the plasma proteomics as a mediator of lifestyle exposures. So 135 exposures across smoking, diet, physical activity, and vitamins, and was able to tease apart tissue and pathway-specific relationship between exposures and proteomics. So things like physical activity had protein expression that was, when looking at GTEx data, inducing response in the heart or in muscle tissue, whereas things like smoking and diet were lighting up proteomic signals that seem to be being expressed in the brain. And an important thing is that these proteomic measurements, when integrating across exposures and genotypes, can be a plausible mediator of disease outcome. So when you ask the question, how much of the exposure correlate to disease outcome, that's a percentage. But then when you ask the question, how do exposures going through the specific causal protein and in fact to the disease, you can get an indirect effect or a mediation of that effect of things that we a priori knew were causally related, proteomics to disease. And layering on exposure onto that gives you a sense of how that pathway could manifest. Amazing data to look at interventions of omics and the response of intervention to omics. So here are a couple of studies. I think Dr. Snyder is coming on later. He's leading studies that look at omics in different behavioral contexts, such as physical activity. There are omics measurements that are emerging through the intervention of the GLP-1 receptor agonists. So you now can compare in experimental settings, human experimental settings, the changes of these omics with the changes of exposure. And that's what we did in our next step. So if you look at proteomic signatures that are induced by individuals who are taking these obesity-related drugs, they're quite similar to those that are emerging from the proteomic signatures that are related to physical activity, and they might even enhance the effect of these drugs. So now we're coming full circle to ask the question, exposures related to disease, but then how that might be related to therapies that are timely and relevant and causal for chronic disease. So if you're interested in this sort of thing, I invite you to join our community as well. We're developing standards for exposomics, is similar to what I just mentioned here, called the Networks of Exposomics in the United States. Here's our website. I'd love to hear from you. Please reach out. I think a collaboration with the Human Cell Atlas seems like an easy win for both parties here. So building an exposome single cell atlas in conclusion here, there's a complex architecture of the exposome from the geospatial to the individual level that impact numerous relevant domains in the blood phenome. And how can we move beyond blood to really get more precise measurements of exposomic function, if you will? So how does this relate to biological pathways and single cell expression? What are the experiments we need to make? I think there's fantastic data already in the Human Cell Atlas on the lung to look at smoking. We can expand this. However, examining the genetic contribution to expression, I think, is easy. Variants are static and non-confounded. And so how do we move beyond transcriptional noise versus true signal of the exposome, and what are the critical study designs we need to make, I think will be very critical for the exposome single cell atlas. So thanks so much for collaborating. Thank you so much for the HCA exposome team and having me speak on this very ambitious but exciting project.
Thank you so much, Dr. Patel. There's unfortunately not time for live question answering, but there is a question in the Q&A, so if you would like to answer that. And with this, we would like to move on to the next speaker. Shyam, will you be introducing her? Thank you, Dr. Patel.
Thanks, Fenna. Next speaker is Sophia George from the University of Miami Sylvester Comprehensive Cancer Center. And Sophia is also co-chair of the Diversity Task Force in the Human Cell Atlas, where she's done important work to facilitate studies such as this. Sophia?
So good day, everybody. And my talk is going to focus a bit on health disparities. And so when I was invited to give this lightning talk, I decided that this was the title, which is Unmasking the Invisible Endocrine Disruptors, Exposomes, and Cancerous in Underrepresented Populations. So thank you so much for the invitation to speak about our work. So there are multiple factors that contribute to health disparities, and I'm also speaking from a US framework, just to give everybody put everybody on the same page here. And so you can see here, this is published from the American Cancer Society a few years ago, where we can see that there are multiple factors, including biology, psychosocial, clinical, but also environment. And work we've been doing now for a while has been looking at different populations, both contextually, where they are in South Florida, but also globally, and comparing this population. So work from my colleagues here at the University of Miami just published last month, that depending on where you live in South Florida, you're exposed to these Superfund sites. And so on the left, they show here that there is a correlation with exposure to Superfund sites and triple-negative breast cancer. And then there's also an increased association with being diagnosed with metastatic disease and being exposed to these Superfund sites. So I have a PhD student that I co-mentor. Her name is Destiny Tiburcio, who's been focused in the lab on studying that actual thing in the context of breast cancer. So we've already heard about these endocrine disrupting chemicals, and there are many of them. They interfere with regular hormonal function, as you've heard. They're ubiquitous. They're found everywhere, including in toys, in cosmetic products, plastics, pesticides, and food packaging. And they also contribute to, we think, certain types of cancers. And so in particular, Destiny's been focused on, and we've been collectively been focused on phthalates. And again, they're ubiquitous. They have some estrogenic activity, and therefore we think can be one of those molecules that will women who live in these environments or exposed to the different factors that I just talked about can increase their risk either for cancer or also increasing the aggressiveness of their tumors. So Destiny's work has been focused on looking at how phthalates affect the circadian rhythm and how that might disrupt the brain metastasis to the brain via disrupting the blood-brain barrier. And this is her active work that's funded by the HHMI. I will just leave that here for now because I want to go into some other exposures that we've been talking about for a long time in the context of immigration. So a lot of our work, as I said, has been focused on different populations, breast and gynecologic cancer, but we've noticed now for many years, just looking at women who live in Florida, initially South Florida, but then now across the state, that we saw differences in outcomes in women based on their immigration status. And what that means is it's really not necessarily the immigration status itself, but the exposures, their genetic factors, their risk factors, and so on. But we happen to be capturing that by where they're born, because people bring their practices with them when they move from one space to another, and also they change those practices. So one of the things that was striking to us when we disaggregated data on Black women who were diagnosed with breast cancer is that we saw that English-speaking Caribbean immigrants had a better outcome compared to Haitian-speaking immigrants and also African Americans at the bottom here. And so my students in the lab and we hypothesized that there's something about this experience or exposure that might mediate these outcomes in particular in these two populations. So what we think that could be. So Haitian immigrants have a difference in language proficiency in the context of English in the United States. There's a fear of deportation, so there is the stressor. They have to navigate a lifestyle, health systems, but also they also have a decreased access to healthcare through insurance. So African Americans, US-born Black women, have multiple types of discrimination exposures, including interpersonal but structural, like redlining. We know that there are food deserts, so access to proper foods. And then there is this chronic socioeconomic stress that we think can contribute to differences either in disease outcome or risk. And then again, as I showed you, where they live. And then you have Haitian natives who are in another type of stress and exposure, which is a political unrest, which is now chronic, or I would say critical, as has been shown in the news over the last 3 years, prone to natural disasters, and of course, affected, unfortunately, by chronic poverty. So many years ago, we got funds from the state of Florida to simply ask in a pilot study, based on our both clinical data and our hypothesis, that there are differences across these three populations who are genetically similar for the most part. And so we developed this pilot study. And the goal is to identify differentially methylated regions associated with nativity and immigration. And again, immigration as the exposure. And so we had women who were in the US, immigrants, and then women who lived in Haiti, all diagnosed with breast cancer, both ER-positive breast cancers and triple-negative breast cancers. And we did a reduced representation by sulfide sequencing. Because of the brevity of this talk, I will only show a few slides of data. So we wanted to disentangle, again, genetic from environmental. And so we had 3 types of tissues, both normal breast tissue, saliva, which was the easiest thing to collect in these different spaces, and then tumor tissue, FFP. And so you see here that when we are looking at, so HI is Haitian immigrant, HN is Haitian native, and USB is US-born Black woman, we see that there are differences, greater differences between the Haitian immigrants and the Haitian natives than, of course, the women who live in Miami or South Florida, but have different, we would call them enclaves of spaces where they live. However, there's still significant differences in the differentially methylated patterns across these populations. And so one of these things that we noticed is for, in particular, the women who live in Haiti, that they have a more immune, inflamed, and this is using germline DNA, the saliva, than women who are in the US. And so we have, where this work is being written up by Jovanka, where we're seeing differences in both the normal tissue, the cancer tissue, and also just the overall health of the person, again, using saliva as our biomarker for differences in exposures and what might be linked to responses in those tumor tissues to both targeted therapy and standard of care therapy, systemic therapy. So while we were doing this, we realized that this was a static study. We had asked a few questions, of course, important questions about breast cancer risk, but we wanted to understand before these women get cancer, before the population gets cancer, what are their exposures beyond genetics, which is what we were really initially focused on. And so we got funding in 2024 from our institution to enroll and to develop this cohort, and we called it the Florida Cancer Health Disparities Study. But within it, we have a sub-cohort called the Santé Nu, C'est Avenir Nu study, which is our health is our future. It's available in English, in Haitian Creole, and in Spanish. But we decided that we wanted to ask these questions about exposures in our Haitian immigrant population and the African-American population. And it just happened that at the time that we launched the study, there was a shift in even their immigration status. So which was a bit, for us initially thinking that we wouldn't be able to enroll individuals. So you can see this, the members. I thought it was important to start there of who is helping us do this work and do it collaboratively in the community. So we're enrolling individuals in the community. Drovanka and Maurice, MD, PhD and MD, who is Haitian. I'm doing his PhD in prevention science. And then we have a clinical oncologist, medical oncologist, who is also recruiting first in the clinic here with women diagnosed with breast cancer. We've done hundreds of different types of community outreach activities, both for just for information and also for enrollments. What are we asking? So we have a long survey instrument. For those of you who are part of the HCA, you heard us talk about tier 1, tier 2, and tier 3 types of data, metadata. I think we're maybe at tier 4, but we ask, as you can see on the left, demographics, family history, of course, medical history, and importantly, environmental and occupational exposures, which include, for us in South Florida, people who are on farms and exposed to pesticides, sun exposure. We have someone who is really interested in looking at hair product use and increased risk for both breast and endometrial cancer, so that is there, and length of time, and also oral health and the social environment. So within this sub-cohort, our goal was to enroll 150 healthy people who have no cancer, history of breast cancer in this case, and 150 individuals who have a history of breast cancer. So this is, again, preliminary data. I'm not showing you any UMAPs here, but this is what we have been able to do is both enroll these women and a few men and women in the non-cancer cohort, and mostly women, but a few men in the cancer cohort. Collected white blood cells, obviously plasma and serum, and in some instances when newly diagnosed individuals collected their tissues, and of course. So what we did with funding and partnership with the Chance Recovery Initiative and 10X and Scale, we were able to generate 268 PBMC single cell maps, and also everyone who is part of the studies undergoing whole genome sequencing at both between 1 and 5x, to be able to, as you heard in the previous talk, start having some more detailed relationships information between the exposures and the self-reported exposures and the transcriptomics of the blood as a way to measure risk in people who yet don't have cancer and being reported to us. There is obviously opportunity to use the plasma and the serum that we've banked to ask to measure in the blood these specific molecules that we think are maybe contributing to differences in the population. So as you know, I was asked to give this talk. I thought about, well, what else are we doing? So we're not necessarily capturing the extended survey instrument. It's an hour and a half. We do it over 2 to 3 visits with the individuals. We have a study that I lead called the African Caribbean Single Cell Network, where our goal is to enroll 3,500 individuals across these spaces where you have the dots, and possibly Brazil now. And so we will have the opportunity to be able to actually, once we're done, because we're banking these tissues, to ask questions about the exposures in an objective way using the plasma that we're collecting, and also obviously the tissues from these populations. So with that, I'll stop. There's a lot of people to thank. It's a big project that we are doing both in South Florida, but also in different spaces. We have great collaborators with the Sylvester data portal system, where we are essentially all the data is being stored to be able, I'm working with data scientists to be able to help us disentangle these different relationships. And our clinical colleagues are helping us recruit in the clinic, and obviously my colleagues across the globe who are supporting this work.
Fantastic. Thank you so much, Sophia, for overviewing these amazing cohorts. In the interest of time, we're going to skip Q&A for now, but hopefully we can come back in the panel discussion. And I'll hand over to Fenna for the next speaker.
Yeah, thank you. So I'm pleased to introduce to you Dr. Michael Snyder from Stanford University, and his exposome talk will be about personal exposomes and health. Mike, the floor is yours. Thank you.
All right. Well, it's a pleasure to be here from dark California right now, I guess. Let's see. And as you heard, I'm going to talk about personal exposomes. It'll go beyond endocrine disruptors. It'll talk about, in general, our work, but again, with the emphasis on personal. Okay, just try to get this. There we go. All right. So I have many conflicts, so you can ignore everything present if you like. And here we go. So I think we all know that your health is influenced by many things. Your genome is important, but it only counts for about 16% of your lifespan. Instead, all these other things, activity, lifestyle, and exposures account for the other 84%. And so there tends to be a lot of emphasis on this, but these are probably equally, if not more important, again, for people's health and lifespan. And so a number of years ago, many of you know, we set up this concept of doing very, very deep data profiling on people where we'll collect blood, urine, and stool, and then sequence their genome once. And from the blood, we'll measure as many molecules and urine as possible, and also study microbiomes and such. One aspect is to collect very, very deep data on people, and the other aspect is to do it over time. And so we've been running the study now almost 13 years. And actually, I'm one of the members. We've been doing over 15 years on me. So, and the goal of this is to try and understand, what does healthy profiles look like, how to change over time, and how can these advanced technologies be used to better manage health? And sure enough, we can actually uncover a lot of conditions pre-symptomatically by actually doing these deep data profiles. Now, the one area that we always thought was missing when we launched this project was actually, what are you exposed to? And so we really wanted to dive into this a little more deeply. And so we decided to look at both biological chemical exposures. Obviously, biological exposures are important for allergies and asthma, chemicals for all kinds of reasons, carcinogens, effects on endocrine disruptors, etc. And we started with some very, very basic questions. What constitutes the personal exposure? What are you breathing right now? And we did focus, I should point, on airborne exposures. How dynamic is it? What's the relationship between the biologicals and chemicals, say, on the outside as well as on the inside? How do these things affect your health? And so what we did was we adapted at the time an exposometer from RTI, where basically this is an air pump. It sucks up, does measure a certain amount of things in real time, sucks up air. You capture particulates like pollen, bacteria, fungi on a filter. And we have a chemical absorbent underneath it to capture all the chemicals. And we do this, we'll sample. You carry these around. Here's the latest one. This is much more sophisticated, much smaller, and it sucks up a lot of air. And it basically, in real time, you can measure PM 2.5, PM 10, temperature humidity, but offline you can measure all the biologicals by eluting what you've captured and sequencing deeply. And you can measure the chemicals by actually doing mass spectrometry and then do a lot of analysis. And so we started this, as I say, a number of years ago. And in the first study, we had about 17 people we were collecting from at various sites. And again, the goal was just to get a feel for what does airborne exposure look like. We had a few people who taken many, many samples. So this is me. I take samples. I've been doing this, I don't know, 10 or 12 years now, collecting literally over 1,000 samples at this point, collect one during the week, one on the weekend. And every time I go to a new location, I'll take one of these with me and sample there. Another person did it for over a year, and this person did it for several months. And so we collected these from all around the globe. I travel a lot. And so we had these. And then what we discovered, I'm going to start with the biologicals, is that, there's a highly diverse set of biologicals people are exposed to. There's about 300 species we'd pick up per sample from relatively deep sequencing, excuse me, and over 2,500 species in an aggregate, including, sorts of things you would expect, pollen, bacteria, fungi. And then we can start diving in and saying, well, how dynamic is the exposome? And the answer is pretty dynamic if you at a personal level, meaning this one's me. These are yellow as fungi or brownish, whatever you want to call that. Red bacteria, green is plants, and bacteria are blue. And you can see that these are different samples collected over time. You can see the different exposures. They vary quite a bit. Same is true for this person, and so on. And we can cluster them so that, these are the plant exposures, these are bacterial, fungal exposures. In terms of number of reads, tends to be the most. And it's fairly consistent. These are my Monterey samplings. Every time I'm in Monterey, I get a fungal exposure that's still held up, and so on. So then we can dive more deeply quantitatively and say, well, where are these? What's the strongest driver of your exposure? And the answer maybe isn't a big surprise, but it's location. So geography is the number one area. This is just 4 people sampled, all in the Bay Area, but they're sampled. They live at different locations. Red is me. I travel, as I mentioned, so my samples are a bit scattered. This person's in San Francisco. This one's in Sunnyvale, which is a few miles south of me. This one's lives and works across the bay. And you'll see everybody has a different exposure, even when they're in roughly the same area, just a few miles apart, makes a big difference. We think, in fact, this idea of measuring exposure at a city level, it's fine at some level, but it's not that high resolution. It misses a lot. And so you can capture all kinds of fun information. So for example, in San Francisco, you'll pick up exposures of these biologicals, including sludge bacteria, which is kind of interesting. If you've ever been to Market Street in San Francisco, you'll understand why that's the case. Smells a bit there. So you get the idea that you get very distinct exposures depending exactly where you live. And then we can quantify this a lot more. And the net result is that location, which is indicated here, is the number one driver. There are strong environmental effects. In fact, seasonal effects are very strong. I'll show you an example of that. There is some technical noise. And a lot of it is just very personal as well. So here's some examples of seasonal effects. These are species that the green one's pink in spring, blue in summer, red in fall, and dark blue in winter. And you can see there's a lot of known stuff in here. Pine peaks in late spring, kind of April, Mayish, which is what we would expect. And here's a fungus that tends to come up in the fall. So you can pick up these things. These are known already, but then there's tons and tons of new ones that, as far as I can tell, have not been described that you can pick up by doing these personal exposures. And in the end, as I'll talk about correlating with health, I have mild allergies, and it turns out it correlates with eucalyptus. I always thought they were pine, but this study told me they're eucalyptus. We can also look at chemicals. We think based on the patterns, there's over 3,000 chemicals people are exposed to. Most chemicals are not annotated, meaning we don't know exactly what they are, but we can guess what their classes are. And we pick up all kinds of classes of molecules. You can see them here: pesticides, carcinogens, plastics are in here, all kinds of fun stuff that you can pick up from, again, these personal airborne exposures. And then you can start, again, looking at how they're distributed. And they are also, just like the biologicals, affected by location. So these are plastics. These are other things. And these are a series of my samples. And basically, every time it rains, you may know you can smell when it rains, because this molecule, geomosin, comes out of the ground. You can smell that. These are the rainy days here. And they basically pesticides, we think also a body scent, maybe because you're inside. You can also pick up plastics we think are getting pushed into the earth. So anyway, this is kinds of correlations you can make. And then you can do all kinds of fun correlations. So you can start correlating on the outside, both biologicals and chemicals, and there's tons of correlation. So yellow are all the chemicals. I won't have time to go through these. I'm just giving you a flavor for the kinds of things you can discover. These are actually the biologicals, and you can see which ones co-correlate by looking across hundreds and hundreds of samples. And some of the strongest drivers of these correlations are the plastics and things like this that are up there, and that microplastics now have a very profound, well, they've gathered a lot of interest, I'm sure you all know. And the other big drivers of these health-related fungi, you'll see a lot of correlations with those, so that get picked up with this sort of thing. And then same thing, we can start correlating biologicals with chemicals. Again, this is all external. And one of the interesting correlations is pyridine, which used to be in paints. It's still found in lots of stuff. It's inversely correlated with fungi. So wherever you have a pyridine exposure, you tend to have less fungi. So here's less pyridine, more fungi, for example. Here's more pyridine, less fungi. And my house was painted by a green guy, no pyridine in the paint. So I get a fungal exposure every time I go home. And yeah, so that's the kind of thing you can discover. And if I were allergic to molds, I might want to have my house painted with the pyridine paint. Probably die of cancer, but at least I wouldn't be sneezing all the time. The other thing you can do is we can start then correlating with what shows up on the inside. Because we profile people's blood and things like this, you can start correlating the internal compounds. So step one is what kinds of compounds are found on the inside in your blood. And it turns out we picked up, this is done with a pretty old mass spectrometer at the time, about 67 annotated compounds, if you will. Only 11 of the same ones were on the outside, meaning you can pick up things probably from food and other sources or things we just didn't have enough sensitivity on that you could, again, find on the inside. These are the classes of molecules. And you can start correlating some of these things, again, with their amounts. And then you can start correlating what's on the outside with what's on the inside. And I'll go through this a little bit quickly, but these are your gut microbiome on the inside here. And these are the highly connected ones that are connected to your external exposures. And again, purple is organisms, and chemicals are in orange. And you'll see there's a ton of connections between your gut microbiome and both chemicals and biologicals on the outside, meaning your external airborne exposure does have a strong correlation with your gut microbiome. And when you start digging in, this is complicated, but the summary is pretty simple. That is, a lot of the chemicals that are dominating this are, in fact, pesticides, plastics, and so on. And they tend to correlate with a lot of inflammatories, so mostly positively correlated. So things involved in pro-inflammatory bacteria in your gut are positively correlated with pro-inflammatory things on the outside and some of these chemicals. That's the upshot. And similarly, we can correlate outside things with your proteome and your metabolome. I won't show all this. And once again, your immune pathway winds up having a lot of the most connections with external exposures, both biologicals and chemicals, and we can pull out exactly which ones they are with this kind of analysis. So again, we can start, these are all associations. We didn't do causal inference at the time, but that is a useful thing to do. And then one thing that interests people the most perhaps is, well, how does this relate to health markers? And so we've done some of that. And as you might imagine, there are correlations between important molecules like glucose levels, believe it or not, correlate with both biologicals and chemicals. So we can pick up some of that here. These are the ones that are significantly correlated. Eosinophils, which are involved obviously in immune response and allergies, they'll correlate with certain external bacteria And similarly, urea and nitrogen we can correlate. So this is the goal. It's really to try to match people's external exposure with what's going on inside, both what we can detect as well as how it impacts your health. And so, in summary, what I've told you about is we have a way of tracking personal exposures. The exposome is vast, both biologicals and chemicals. The exposome is very dynamic and is largely driven by location, and season is a strong factor. There are lots of correlations between chemical and biological patterns, mostly location-based, as shown here. And again, we can correlate what's on the outside with inside things, including health markers as well. And these are the folks who did the work. A very talented team over the years has worked on this. This is a group that started the project. And these are some folks who did it more recently. Peng did a lot of the correlation work along with Xiao Tao. And our guru experimentalist is Allison Zhang. So that's the summary. Happy to take questions if people have any. I hope it wasn't too fast.
No, this was great. Thank you so much, Dr. Snyder. For the interest of time, we are moving the questions to the more general panel, where we will be exploring ways to strengthen bridges between the Human Cell Atlas and the Global Exposome Forum towards the Human Exposome Atlas. So Shyam will be moderating this session, and if everybody can put their cameras on for the panel, then we can get this panel started.
Thanks, Fenna, and thanks so much to all four speakers of our session just now. Nicolo, Chirag, Sophia, Mike, for really setting the stage and showing us the power of this field and some of the possibilities. I know Chirag has to leave soon, so I'm going to start with him. Chirag, nice to see the analogy from GWAS to exposome as your predictor variable. And you've heard Mike's talk also just now about at a fine scale what all the airborne and other exposures are so diverse, right? And you also cataloged a lot of exposures. So, but you correlated them to phenotypes, which is really important, which brings me to my question of if these two consortia are to come together, Human Cell Atlas and Exposome Forum, what are some of the big questions they could tackle? And by a big question, I would code that as what are the major exposures we really need to be looking at, and in the context of which phenotypes?
Absolutely. So I think that the exposure-wide analysis paradigm could help one prioritize exposures from the epidemiological level. So I think those are themes that were shared between all the talks, is that you can get to some sort of clinical outcomes by looking at the population, and then start to look at those in the single cell context. So where I think that the single cells analyses or assays are most important is helping us get to how exposures impact biological pathways. That's our win. We're right now studying exposures in blood or in convenience samples, but really mechanisms occur in a tissue specific way for different diseases, right? So I would say that it can be done in a tiered fashion. You start with those exposures that are prioritized from existing studies or exposure-wide analyses, and then you move those into the single cell context.
Thanks so much, Chirag. I was wondering if you could stick your neck out and nominate like some particular exposures and some particular phenotypes that you feel that if we were to address them scientifically and then translate them, they would make an impact on public health. And I know that's like a lot of crystal ball gazing, but why not?
No, absolutely. So I think that so first off, I think we can prioritize the dietary biomarkers. I think in our atlases that we've been creating, those have a super strong relationship on an individual level with across metabolic phenotypes. So things like glucose, things like insulin levels, adiposity. I think we see that across the board. I think the critical questions there for the field is like, how does diet and physical activity interact to induce response in those metabolic factors? And I think that's, where the personal sensors come into play. So I'd say you'd have to start with diet to help us resolve some of the outstanding questions that we have about dietary exposures. Others that are emerging in our atlas include those that are prioritized by the CDC. So we see that blood parameters such as red cell distribution width or white cell count, immune parameters have a relationship with PFAS, and these are like compounds that are been emerging in the popular literature lately, just because they're omnipresent, and there's so many of them, and they seem to be inducing response in those immune related parameters. And I would say that those are also important because there is some evidence to suggest that they interfere with vaccination. I think there's a paper in JAMA maybe 5-6 years ago showing how that occurs. Other exposures that are emerging include heavy metals, and those have been mainstays for indicators of both socioeconomic status, but also adverse response to things like Alzheimer's disease. I showed a slide earlier in that New England Journal of Medicine case study where these heavy metals are accruing in the brain. I think that's a great case study to understand tissue-specific response of metals in neurodegeneration. So I think in summary, you think of diet, first of all, you think of exposure-wide studies as a way of prioritizing. But in those atlases that we've been developing and kind of cross-referencing with the personal exposures that Mike is developing, you see that those that are prioritized by the CDC, like PFAS, metals, and dietary markers are definitely emerging and coming up short second are phthalates across things like metabolism and brain health, brain aging. So is that a bit of my neck out there?
You certainly did all the way, and truly appreciate that. A great starting point for the road mapping exercises we will come up with between the GEF and HCA going forward, and hope you will be a key part of that. But I'm going to zoom out a little bit because we have the architects of these two consortia and the person who brought us together on this panel. So I'm going to go to them and then come back to the science. So, but hoping Casimiro and Fenna and John can do some framing over here. They've already introduced the topics. Maybe Casimiro, you could say, as the host for this, what would UNESCO love to see from these two consortia coming together that would align well with the goals of UNESCO?
Absolutely. Thank you, Shyam. As we close, actually, I would like to underline one simple idea. A Human Exposome Cell Atlas is not only a scientific project. It's a choice about the kind of multilateralism we want for biomedical sciences. And if we succeed, it will be because laboratories, ministries, funders, and communities have decided to share data, methods, and responsibility in a spirit of open science, equity, and trust. What UNESCO will do, UNESCO will continue to provide a neutral platform where these different worlds can meet, negotiate standards, and keep people and planets at the center of the discussion. I would like to thank all the speakers and the participants for the clarity and the ambition you brought today. UNESCO is really committed to create synergies and as you know, we are in close contact through our permanent delegations to member states. So we are the voice to transform science and to translate science into policy and to support this kind of efforts for the scientific community and for the benefit of everybody. I hope that when we meet again, we will not only be presenting the results, but also new partnerships and capacity built in parts of the world that are still underrepresented. This is extremely important for us at UNESCO. Thank you.
Thanks so much, Casimiro. We will come back to that topic of underrepresented communities and global reach very soon. And definitely important for HCA and GAF as well, a key objective. Fenna, coming over to you, perhaps you could talk about you take all this science and then how will it map to actionable, say, exposure thresholds, clinical pathways, public health policy, and so on?
Yeah. So there are different layers in that aspect. Some of our speakers already indicated layering at the level of pathways, mechanistics, so the biological level in a sense, either at the individual level or at the population level. And then what the Global Exposome Forum tries to do with working towards the Human Exposome Project is to put this at the bigger scale where we can find the common denominators within these studies so that we can have either reference or, although the challenges of reference human exposome has been discussed in previous discussions, but some sort of consensus of how to assess the exposome for the human population or the human individual. And that then translates to either precision medicine, and Dr. Snyder has examples of that, where that knowledge is captured. So how does that exposome contribute to one's health? But also there are discussions at the city and state and country level where countries are considering the population health. What is the exposome contribution to that health status? And so policymakers are very interested in this as well. And that's what the Global Exposome Forum, and we chose the word forum on purpose because it brings different stakeholders together, is where the Global Exposome Forum tries to be that facilitator of having things that come out of academia and from the bench, from the field, how that translates to policy or implementation, translation. So it could be policy and implementation at the state population level or in the clinic with precision medicine, for example. So this is where the Global Exposome Forum tries to bring those groups together.
Fantastic. Thanks so much for that. And excellent point that to get the attention of policymakers, we need to show the impact, how much people are exposed to and what they're exposed to and how that modifies outcomes, how that influences
Yes.
outcomes. And if we can show the magnitude of the effects and the specificity of the effects, then some policy changes are feasible, or at least we can seed that conversation. John, wanted to come over to you as really one of the pillars of the Human Cell Atlas and someone with visibility into how these consortia function. It's a question, given how important this topic is. I think one question on people's minds would be, why has all this not happened yet? Why has these genomics consortia and Exposome Forum, other such consortia not come together yet to work together? But instead of phrasing that as a negative question, I want to phrase it as a positive question, which is, what does it take to make this kind of community happen? In all respects, from the funding respect, from the consortium aspect, ethics aspect, everything, right? To make such large scale projects possible. Big question, I know. Take whatever you want from it.
It's also a great question, Shyam. And I think what I've just seen over the years that I've been with HCA is it's often a question of having a vehicle for connecting researchers, and that's why I'm excited about this collaboration. I think all three of our organizations have the goal, as you've heard, of finding ways and organizing activities to help connect researchers. What we often find in HCA is that at our meetings, we'll see connections form between researchers who hadn't worked together previously or may not even have realized that they worked on a common topic. And so I think to the extent that we can continue to organize these types of activities to promote collaboration, that would be fantastic. I think another area that we as HCA want to support has already been mentioned, but it's really around standards development, around metadata standards and so forth, to enable the interoperability of data and to really sort of facilitate collaborations even around data that's already been produced. And then I finally would say that we've seen success in areas like disease studies in forming new collaborations that then can be much more competitive for research funding. And that's been true at the regional level with some of our regional networks on various continents, but also just within tissues or groups of tissues. Bringing together researchers is often a really great way to sort of increase the strength of a research proposal and enable additional research funding to be produced. And so I'm hopeful that through this collaboration, we'll also find similar opportunities.
Thanks very much, John. Indeed, you hit upon one of the key pillars or key foundation of Human Cell Atlas is a shared single cell resource that everyone contributes to and everyone accesses. And surely that is a good paradigm for coming together with the GEF as well. We'll have to conceptualize new kinds of data resources, perhaps tissue bank and metadata resources, and then also standards that, as you mentioned, so that researchers across the consortium can work in a harmonized way so that the studies can be comparable across sites and across teams. And finally, you mentioned funding and perhaps related to the question of how do we form specific teams around specific diseases in the case of Human Cell Atlas, say, or organs in the case of Human Cell Atlas, but in this case, specific exposures, perhaps, or combinations of exposures and diseases. So those are, I'm sure, the ingredients of some such of these initiatives. I know Casimiro brought up the equity and global impact. And so there are quite a few questions in that vein as well. Questions about ethics and return of results, questions around community engagement and LMIC partners. If people don't know, LMIC stands for low and middle income countries. Some people don't like that term, but has been used widely as well. How can everyone be equal partners in this enterprise? And how can we do this research in a way that involves a very broad spectrum of humanity rather than a more narrow slice that for whom science is geographically and financially more accessible? So that's a very large constellation of questions. Again, but I'm having to bundle questions into meta questions because this is a short session. And I'm going to throw that entire bundle over to you, Sophia.
Well, I actually felt that bundle. Hi, Shyam. What can we do? So one of the things, obviously, that we're doing is having this, I think, webinar that's been shared widely through the HCA, which John showed that they're members across many, many countries. It is some places have access to the equipment and they have no resources, not sufficient resources to run the experiments, right? And some have the access to the people and the data and not the expertise to analyze the data or run the resources or to run the samples to analyze them. So I think it's like being aware that there are these disparities in access and training. I saw someone ask about, as a postdoc, which is the best way to go to be able to participate in this type of work. It's continuously engaging our partners in a way that they feel that they are benefiting from the participation and not just giving. I wanted to ask about what do we mean when we want to return results? Like, what are the results that we're returning in the context of exposomes and exposure data? And some people can't move, right? So when we talk about where people live and the exposures, what's the end results? Like, is it going to be a pill that can decrease our risk for one disease or another, changing the paint in our house, or the type of paint? So there's a lot of things that we have to think about in the context of how this work is impacting the people who live on the ground in different spaces, and of course the different types of exposures that they have. So I'm looking forward to the collaboration. I'm looking forward to how we're going to keep engaging our partners in spaces across the world with different resources, and actually the resources that we want to be able to study, so they have that power and leverage, because they have people and exposures that are different and distinct from us here in the US.
Thanks so much, Sophia. Your support and leadership will be very, very important in this. I see Fenna has raised her hand. Maybe a comment on this.
Yeah, if I can comment on this a little bit. With the Global Exposome Forum, we are also exploring this nexus of making it actually global. And for example, we were present at the Exposome Symposium in Santiago, Chile, and a lot of local South American and Central American groups were there presenting their data and discussing their point of view of how to address the exposome and the human exposome project at large. And this is why I said at the beginning, I don't think there will be one human exposome project. It will be regional, because exposures are regional, etc. But that doesn't mean that we can not learn from each other's data and from each other's findings, because certain exposures may overlap, or even though the overall complexity of it, the overall exposure might not, there are ways to learn from each other and what type of exposures in certain settings may inform specific groups or in other regions, for example. So what we learned from those discussions so far, and this is obviously open for further discussions, is that it's important to make sure that regional chapters or regional areas are working on these things themselves as well, and that we have shared data sharing so that we can learn from each other, both on the methods, but also on the data and the findings themselves. And even though, like you said, low and middle income, and Sophia mentioned maybe not all resources are available, there are actually quite a lot of resources available in almost all the regions, because from Irene, for example, they were working very much on exposome infrastructure, and they were indicating that there are expertise labs, maybe in a neighboring country or in another area close by, where you could leverage and collaborate with, so that you don't per se have to collaborate with always Europe or always the US or something like that. So you don't have to per se collaborate with more affluent countries or more resource-rich countries, because within a region, there might be pieces that you need to put together with the puzzle. And I'll leave it with that. So we have to be creative in that sense. But I'll leave it with that. There were many other thoughts on this, but I want to bring it back to the panel.
Thanks, Fenna. We want to get to Mike and Christoph and Nicolo also, but quickly, Casimiro.
Yeah, just to say that I think that how to raise awareness, that I think is the most important thing about this kind of synergies and focusing on the relevance of the research behind. We have to build on the momentum. WHO mentioned that health should be the metrics for climate change, for example, and this is also been said in several. I did was say that COP in Belen. So I think. We need to, and this is why UNESCO is here. We need to create awareness in a way that is that reach policymakers and funders. This is essential to have that fair distribution of the initiative.
Excellent points, Casimiro. Thank you. Quickly over to you, Mike. Really gobsmacked by the depth and breadth of exposures that you're measuring, which brings up the question of if we want to do this at scale, it'll involve a fair bit of funding, perhaps. And you have, as you showed on your first slide, a lot of experience engaging with industry. So that brings up a question from the Q&A, actually, from Jo-Chun Shang. How do we engage industry partners in this kind of research to work with them so that they can bring in some funding and additional resources as well, and then make it beneficial for them too?
Well, good question. I mean, to be honest, industry is motivated by money. So there has to be a market for it if you really want to get down to it. Or you need foundations to step up like Gates and such. So, but having said that, one area that I think is of high interest that I think works nicely in the Exposome HCA interface is you can now do single cell profiling for metabolomics and lipidomics, which means you can measure at a single cell level environmental toxins and things. So a big burning question. So as everyone said, and I 100% agree, we need to correlate exposures with health. And so first of all, we should be collecting health information around all the samples that are being analyzed in general. But now with these new technologies, you can actually, a burning question is what does cell organization look like in the face of certain exposures and such. And so there are companies that can actually measure this to show what molecules are sitting at a single cell level that you could then bring in and see exactly what toxins are sitting in cells that have interesting organizational patterns. And I think that's an obvious interface between HCA and the exposome, if you know what I mean, because tumor microenvironment's a good example. we have millions of tumor cells, all of us do, sitting in our body, but most of them don't cause cancer, but some break out, presumably because the organization has been disrupted or been affected in some fashion, probably a lot by environmental exposures. And we're in a position now with some of these new technologies. And so I think that's an easy win with some of these mass spec companies to try and capture that information, something that's not being captured at all right now. So that's just one area. There are many areas as well. I would say there's just simple things, maybe to play off of what Sophia and others had said. If we can, when we collect samples, let's just also get the environmental information that goes with that. We usually don't do that other than at a very gross level, what city they're from, which is really not that informative when you want to know exactly what exposures are being, showing up with what samples. So I've covered several different topics in my answer there. So I don't know if that's exactly what, I know you want to get more at industry, but yeah, you've got to motivate them. And I hate to say it, but it's much more for foundations because of the nature of the way we fund things.
Absolutely. Foundations would presumably be a great partners for this, but you mentioned biotech industry as well. That makes a lot of sense, and they partnered with the Human Cell Atlas before and would be natural partners too in this case.
Yeah, mostly from the new technology side, right?
Absolutely. Absolutely. A natural match, I would say. We're running out of time, but still, it's a privilege to have also Christoph Bock here, and wanted to get to Nicolo also. I know you're both working on organoids, and so that perhaps we can combine some questions from the chat and also from Nicolo's talk. Very broadly, people are interested in virtual cells and these organoid models with the perturbations that Nicolo was talking about perhaps lend themselves very nicely to virtual cell modeling. But then there's also the ever-present question when you're doing organoid modeling of how do you show that it matches what is happening in vivo as well, which again perhaps brings in Human Cell Atlas. So maybe Christoph, I'll pass that to you.
I think the real challenge here is the kind of essentially the scale of the problem. We have a huge number of exposures and we have a huge number of phenotypes, and we need to connect them. And many of those have to accumulate over a human lifetime to really have an effect. So it's not like an allergen that causes kind of an immediate effect, but it's a slow build up of an exposure. And kind of if you look at that from the eyes of an epidemiologist and you try to do statistical power calculations for that, you very quickly come up with numbers that exceed the human population. So even if we could track every single person on the planet, we would not have the statistical power to really sort out the combinatorics of our exposures as a pure statistical problem. Meaning we need to build kind of pathways of causality or chains of causality where kind of the associations that we get from observational studies, in particular also time-resolved perturbational studies, are connected with causal arguments from in vitro models. And their organoids have a lot of promise because they allow us to look beyond the blood. They allow us to, in particular, study the brain, which is almost impossible to do in any other way, but also many other organs. So I think when we think about standardization, we should also start lining out what we consider sufficiently strong evidence. And such sufficiently strong evidence will often be of a chain of arguments that are partially based on kind of exposome studies and statistical data, but also partially on causal arguments and mechanistic evidence from kind of in vitro exposure models.
Thanks very much, Christoph. That's an excellent point that you need some kind of biological framework and mechanistic insight to go beyond just pure combinatorics and statistical associations, and that might make our life simpler in understanding the effects of these exposures. I don't know, Nicolo, if you want to take in 60 seconds, because we're out of time, the question about can you scale up the models that you're using, and will that benefit virtual cell modeling by exploring the space of states more broadly? Just a quick answer, if you don't mind.
Yeah, I mean, we are investing all our effort in scaling up these models. My only additional consideration is that
we can't hear you too clearly. Maybe if you come closer to.
Can you hear me?
Yeah. Thank you.
Okay, sorry. Yeah, I was saying that we are putting all of our effort onto scalability, and one simple additional consideration for how to explain why I am so excited of the convergence between exposome and human cell atlas is that we are starting to not only understanding what are the problems, but how they act in terms of our exposomics. And I always like to invite people to reason on the unicity of organoid modeling, which allow us to differentiate the same genomes as many times as we want. So for the first time, we can isolate what is the developmental landscape of different genomes. So if we integrate cohorts, we go in vitro and we differentiate them, we can understand really where is the genetic influence on developmental trajectories, and then test how the exposome that we can now start measuring also on a personal level impact on those. So this
Thanks so much, Nicolo. Yeah, I'm sorry, I'm going to have to close the session here. Really stimulating, amazing to have all of your expertise on this panel. Thank you so much for joining. I'm going to hand over quickly to Casimiro and Fenna and John for their closing remarks. Thank you all.
Thank you all. Casimiro, you want to start us off?
Thank you. Thank you very much. I think that is an important element I would like to underline to close is that, of course, thank you to everybody. It was so interesting and there's a first step that is very promising. I think we have to see it in a very, I would like to use this word, the One Health, planetary health approach. And this is basic research on in this sense. And I think it's essential nowadays to to put this at the basic of this approach. UNESCO is here to support, as I said, and I think we will do our best to make, as I said, awareness, to create equal access, and get and translate. Once again, I will use this word because sometimes is what is asked by policymakers and our partners, to translate the importance of what you are doing in a language that can be understood by our member states and partners. Thank you.
Yeah. From the Global Exposome Forum side, we are very excited about this partnership and for this webinar series to bring these different communities together, because that's exactly what we as the Global Exposome Forum intended to set out, is to bring different stakeholders together towards this overarching goal of the Human Exposome Project. And that means translating indeed to science, all the way to policy and to end user, so patients being treated or being assessed by their exposome as well. So not just their genetics, but also their exposome. And we are providing this platform. We are happy to collaborate, and we look forward for additional interactions with these different groups. We've set out to also follow up with this after this webinar with a white paper together with UNESCO and the Human Cell Atlas group, so that we could have this discussion at a more broader level and more public level also, so that the whole society can participate and benefit from our outcomes. So looking forward to additional webinars in this series and towards even broader collaboration with the two groups. Thank you.
And I would just add, as Fenna says, this is really just a start. We really want to hear from you about topics and opportunities for this collaboration and for future webinars, as well as future meetings. I want to thank Casimiro for bringing us together in the first place, and all the speakers for the really wonderful science that they brought to the event today. Thank you.
Yes, thank you. Thank you to all, and especially to Casimiro.
Thank you. Thank you.